Continue our discussion on glycolysis by looking at the molecular level, what happens or the sequence of chemical transformations from glucose to pyruvate. So let's take a look at one of them, at each of them. So initially, uh, glucose is phosphorylated, okay? So you can appreciate here in this figure, the source of the phosphate group is one ATP molecule that's dephosphorylated via the action of a kinase enzyme, and that phosphate group is transferred from ATP to the glucose molecule. The original glucose molecule is now transformed as a glucose 6 phosphate. So the phosphate group is the one I am emphasizing here. Then, with the rearrangement brought about by another enzyme or catalyzed by another enzyme, the glucose molecule is transformed, the glucose 6 phosphate molecule is transformed to fructose 6 phosphate. Okay. This fructose 6 phosphate will undergo phosphorylation via the action of the enzyme phosphofructokinase. So a second phosphate sourced from another ATP molecule is transferred to it, hence becoming fructose 1,6 bisphosphate. Okay, so you can see the molecular changes or the positions or the carbons where the phosphate groups are attached per step or per intermediate molecule. Okay. The fructose 1,6 bisphosphate will now be broken down into two 3-carbon molecules. So you remember the two types or the two byproducts, DAP, dihydroxyacetone phosphate, okay, and PGAL, phosphoglyceraldehyde, okay, or and just to reiterate, okay, DAP is the less stable 3-carbon byproduct or 3-carbon intermediate, so it will not stay as such for a while, for a longer period of time. Instead, it will be transformed, it will be converted into phosphoglyceraldehyde. So you have at the end of the breakdown of fructose 1,6 bisphosphate, you will have three molecules of phosphoglyceraldehyde. This one and this one. Okay. The next step is oxidation and further phosphorylation, right? So a NAD plus molecule or two NAD plus molecules are reduced to NADH. An inorganic phosphate comes into play, or two inorganic phosphates come into play, and your phosphoglyceraldehyde molecules are converted to 1,3-bisphosphoglyceric acid or bisphosphoglycerate. So two molecules of 1,3-BPG. So you can see that on both ends there are phosphate groups that are attached. Okay, so this is a first example in which or an example within the glycolytic pathway where reduction as a key activity during metabolism is exemplified. So moving forward, the 1,3-bisphosphoglyceric acid molecules are further processed under the or are further broken down or transformed through substrate level phosphorylation. So I want you to remember this term, substrate level phosphorylation, which is specific to glycolytic pathway, no? So during substrate level phosphorylation, the 1,3 BPG molecules are transformed to 3 phosphoglycerates. And during that transformation, phosphates are phosphate groups. One of the phosphate groups from each molecule are used to form ATP molecules. So two ATP molecules are produced during substrate level phosphorylation from a single glucose molecule. These three phosphoglycerate molecules later on rearranges to form two phosphoglycerate. Okay, so phosphate group transfers to carbon number 2. And parallel to that, there is expulsion or release of water molecules. Okay, um, the two phosphoglycerate intermediates will then be transformed as PEP, phosphoenol pyruvate. Okay, so the phosphoenol pyruvate is like a mirror compound to phosphoglycerate. And lastly, before it produces pyruvates or pyruvic acid, okay, um, it will give off its they're the two PEPs. They will give off their phosphate groups. They will release it, and via after releasing or um, the release of phosphate group is in conjunction with the another substrate level phosphorylation occurring before the ultimate step of pyruvate production. So two ADP molecules are phosphorylated as ATP, yielding or allowing the transformation of PEP to pyruvate or pyruvic acid. Okay, so you can see the molecular changes at the compound level. Okay, now just to reiterate how important these reduction and oxidation reactions are within the glycolytic pathway, throughout the glycolytic pathway. So, um, you examine or you remember the difference between them by looking at the donor molecule. So the recipient molecule. So the recipient molecule is the one that is either reduced or oxidized. So in this example, we're seeing, we're showing pyruvic acid transformation to lactic acid by giving off hydrogen, by giving off electrons. Okay, pyruvic acid transforms to lactic acid, a reduced form of it. On the other hand, as another example, we have the formation of acetyl groups or acetyl group from pyruvic acid. And here, what we are seeing is the acetyl group is actually oxidized in comparison to our other example a while ago, where pyruvic acid was transformed to lactic acid. No? So this one happens within the mitochondria, while the other one I've shown a while ago is happening within the cytoplasm, where oxygen is not available, not readily available. So, okay. Okay, as an example, the most, I'd say, the most prominent example within the glycolytic pathway that shows us the importance or the significance of reduction reactions is a transformation of NAD to NADH. No? Okay. So just a summary of everything, to high level summary. The glycolytic pathway has two major stages: an investment and a payoff phase, where ATPs are consumed and produced respectively. So please do remember the steps during each or for each stages, because they will be asked specifically during our examination. Uh, the enzymes, please take note of the different enzymes that are involved during the transformation of glucose or transformation of glucose to pyruvate or pyruvic acid. Okay. Now, some of the important features of glycolysis that we should not forget, okay? So, in terms of the key transformation, one glucose molecule yields two pyruvic acids or two pyruvates from one six carbon compound to two three carbon compounds. By nature, glycolysis is anaerobic, but the succeeding steps, of course, will be dictated by the presence or absence of oxygen. So, in the absence of oxygen, the pyruvates cannot be translocated to the mitochondria. They cannot be used for oxidative phosphorylation, and the ATP yield is much lower compared to oxidative phosphorylation, where the pyruvic acids are transferred or translocated to the mitochondria because of the presence of oxygen. Now, in terms of the energy net gain, the end product is two ATP molecules. And of course, there's also the development or the production of NADH, reduction of NAD to NADH as important carriers of electrons that can later on be utilized during the citric acid cycle, the electron transport chain within the mitochondria. Generally, glycolysis serves as a backup for energy. So, in cases where oxygen or the cell is deprived of oxygen, and of course, it still needs to make use of ATP molecules, glycolysis can occur or glycolysis happens. No? In tissues that are highly oxidative, pyruvate production is very important for oxidative phosphorylation, for acetyl CoA synthesis. No? And in conditions where oxygen is not involved, glycolysis relates to lactic acid fermentation. So it happens prior to lactic acid fermentation because pyruvate is needed for lactic acid fermentation. In animal cells with little to no mitochondria or those that do not have high metabolic requirements, non-oxidative tissues or non-oxidative cells, lactic acid fermentation is actually the primary source of energy, primary source of ATP. In the animal body, specific locations where anaerobic glycolysis is important or is involved include the eyes and the skeletal muscles. Now, the last part of our recorded lecture is the regulation of the glycolytic pathway. So there are several things, factors that are involved, dictating the speed of the catalysis process or the speed of the catalysis processes because there are different steps where different enzymes are involved. So first is the amount of glucose. So definitely the lower the amount of glucose available, then the slower glycolytic pathways are slower glycolysis um, proceeds. No? The amount of oxygen definitely, as I've mentioned, in there are two possible fates for the end products of glycolysis. So oxygen presence or absence dictates which fate will be realized more likely than the other. Then we have allosteric regulation, where enzymes, okay? So enzymes are regulated based on the presence or absence of the substrates that they process. No? Later, I'll show a representation. No? And then transcriptional control at the level of the RNA, okay, before you have the enzymes that carry out the glycolytic steps. So enzyme induction. So in instances where the transcriptional regulation of the RNA coding for the specific proteins, the specific enzyme proteins involving each step of glycolysis. If regulation occurs at that level, let's say that the mRNA strands are controlled or are repressed, they are not allowed to be produced, they are not allowed within the cell, their activity is dampened in a way. Okay, so that particular situation actually equates to glycolytic pathway being so slow, no, or being slow than the usual one. So as an example for enzyme regulation, I mentioned a while ago that the key enzymes, specific molecules can inhibit or activate them depending on the concentration of these molecules. But the idea is simply when the byproduct when the byproduct of the enzyme catalysis of a specific substrate is abundant, the activity of that enzyme needs to be controlled. And on the other hand, if the byproduct is low in concentration, the activity of that enzyme should be activated. That's the entire simple idea on enzyme regulation in relation to glycolysis regulation. Okay? So in our on-site lecture this coming Saturday, I'll be discussing um, the I'll be discussing oxidative phosphorylation, electron transport chain, the processes that happen within the mitochondria.